Discussion 7, group members Citlali, Damaris, and America.
I selected DNA polymerase for my enzyme. So DNA polymerase is an enzyme that carries out synthesis of DNA molecules from deoxyribonucleotides. The most important task of this enzyme is to copy the genome when a single cell splits into two. How does it function? The mechanism through which DNA polymerase copies a DNA strand is to add on to the existing strand by reading its sequence and adding on complementary nucleotides to create a new 5 to 3 strand. DNA polymerase also has proofreading functions, enabling it to check for errors during DNA replication, making it highly reliable in DNA synthesis. DNA polymerase was first discovered in 1956 by Arthur Kornberg, a molecular biologist who studied the enzymes responsible for DNA replication and the repair in the bacteria E. coli. By isolating this enzyme, Kornberg and his colleagues established that DNA polymerase 1 can synthesize DNA in vitro. And Kornberg was awarded the Nobel Prize in Physiology and Medicine in 1956, and his son, Roger D. Kornberg, had also made important discoveries about transcription and replication process in DNA. And then DNA polymerase are found in virtually all living organisms, including plants, animals, and humans. And then DNA polymerase are found in the nucleus and the mitochondria of cells. DNA polymerase is essential to life because it ensures the accurate copying of genetic material during every cell division. And DNA polymerase increases the speed of DNA synthesis. Without it, the speed of DNA synthesis would be drastically slow, and so cellular division and the propagation of genetic materials would be very insufficient. DNA polymerase would ensure that DNA synthesis is fast and accurately carried out. It is important for the growth of all of organisms and its development and maintenance. And that was mine.
Sorry, I'm trying to share my screen. Okay, can you guys see it?
Yeah.
All right. What is the enzyme and what does it do? Enzymes are proteins that act as biological catalysts by accelerating chemical reactions in our bodies. Lipase is an important enzyme in the digestive system that helps break down fats into smaller molecules called fatty acids and glycerol. The process is essential for the body to absorb and utilize fats from the diet. Lipase is produced mainly in the pancreas, but also found in the stomach and mouth. By breaking down fats, lipase ensures that the body can effectively obtain energy and essential nutrients from fatty foods. When and how was it discovered? Lipase was discovered in the mid-19th century, around 1840s, by Claude Bernard, a French psychologist. Bernard conducted an experiment on pancreatic juice and found that it contained enzymes capable of breaking down fats. His work laid a foundation for understanding the role of enzymes in digestion. By isolating and studying lipase, Bernard was able to demonstrate its crucial function in the digestive process, paving the way to the future research into enzyme activity and digestive health. Bernard's groundbreaking work demonstrates that enzymes' ability to break down fats establish crucial roles in digestion. Numerous scientists have contributed to the understanding of lipases. In the early 20th century, research like John Northrop and Wendell Stanley advanced the field crystallizing enzymes, including lipase, which helped elucidate their structure and functions. These efforts have paved the way for the modern biochemistry, enable a deeper understanding of enzymes, mechanisms, and their applications in medicine and industry. It is Is it found in human and other biological organisms? Yes, it is found in both humans and animals. It has the same essential functions. Lipase is present in the mouth, pancreas, and stomach. If found in the humans, it is found in mouth, pancreas, and stomach. How does the enzyme contribute to life? Enzymes are proteins that act as biological catalysts by accelerating chemical reactions in our bodies. Essential for digestion, liver functions, metabolism, and DNA replication. Yeah, and that is mine.
Nice.
Okay, mine is okay. Mine is amylase. Amylase is an enzyme that is primarily secreted in the pancreas and the glands that make saliva. The primary role of amylase is to perform hydrolysis by breaking down the glycosidic bonds within starch molecules, which consist in long chains of glucose molecules, converting complex carbohydrates into smaller sugars, making it easier to digest and absorb carbohydrates by the body. It was initially discovered as diastase, now known as amylase. It was one of the first enzymes to be isolated and to undertake scientific investigation by French chemist Anselme Payen. However, in 1908, a study by Wohlgemuth identified the presence of amylase in the urine, paving the way for its use as a diagnostic laboratory test. There are three main classes of amylase: alpha, beta, and gamma. Each one of these targets a different segment of the carbohydrate molecule. Alpha amylase is present in humans, plants, animals, and microbes. Beta amylase is found in microbes and plants only. On the other hand, gamma amylase can be in both animals and plants. One of the ways that amylase contributes to life is by its use for diagnosing pancreatic diseases, and it's a frequently measured enzyme owing to the accessibility of cost-effective and readily automated methods. Although amylase is a sensitive marker for acute pancreatitis, its lack of specificity is evident, as it can be elevated in numerous conditions unrelated to the pancreas.